Cellular respiration is the process of making ATP using glucose and other nutrients. Cellular respiration can be either anaerobic, without oxygen, or aerobic, with oxygen. Glycolysis is the only step in cellular respiration that is anaerobic. Pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation all require oxygen and are therefore aerobic. There are 4 steps in cellular respiration. The first step is glycolysis, which is the breakdown of glucose into 2 molecules of pyruvate. Glycolysis can be broken down into 2 stages. The first is an energy investment stage, and the second is the energy payout stage. The second step of cellular respiration is pyruvate oxidation. This results in the generation of carbon dioxide and 2 molecules of acetic-O. This occurs in organelles that may be familiar to you, the mitochondria, you guessed it, the powerhouse of the cell. The third step in cellular respiration is the citric acid cycle, followed finally by oxidative phosphorylation in conjunction with the electron transport chain. Glycolysis occurs in the cytoplasm of the cell, while the rest of the stages of cellular respiration occur in the mitochondria. The molecules generated through stages 1 through 3 are utilized during oxidative phosphorylation to generate the majority of the ATP that is created. Going over glycolysis in deeper detail, we see that in the first half ATP is used by hexokinase and phosphofructokinase in the generation of 2 glyceraldehyde-3 phosphates. In the second half, GAPDH generates 2 nicotinamide adenine dinucleotide molecules and 4 molecules of ATP. After subtracting the 2 ATP from the energy investment stage, the net yield is 2 molecules of ATP at the end of glycolysis, with the generation of 2 molecules of pyruvate. The 2 pyruvates are then converted into acetyl-CoA, and during this process carbon dioxide is released and nicotinamide adenine dinucleotide is produced. The acetyl-CoA molecules are combined with oxaloacetate and progress through the citric acid cycle. During the citric acid cycle, succinyl-CoA is involved in the generation of ATP. The final product of the citric acid cycle is oxaloacetate, which can be recycled and reacted with additional acetyl-CoA molecules. Nicotinamide adenine dinucleotide molecules and FADH molecules are used in the electron transport chain to create a hydrogen ion gradient with 4 complexes. ATP synthase is an integral membrane protein that harnesses the energy of the proton motive force by allowing hydrogen ions to diffuse down their electrochemical gradient, causing components of this protein to spin, making ATP. Hydrogen ions in the matrix space can only pass through the inner mitochondrial membrane by an integral membrane protein called ATP synthase. This complex protein acts as a tiny generator, turned by the force of the hydrogen ions diffusing through it, down their electrochemical gradient. The turning of parts of this molecular machine facilitates the addition of a phosphate to ADP, forming ATP using the potential energy of the hydrogen ion gradient. The theoretical maximum yield of ATP made during the complete aerobic respiration of glucose is 38 molecules, with 4 being made by substrate-level phosphorylation and 34 being made by oxidative phosphorylation. ATP is often called the energy currency of the cell. ATP is generated through 2 mechanisms during the breakdown of glucose. A phosphate group is removed from an intermediate reactant in the pathway, and the free energy of the reaction is used to add the 3rd phosphate to an available ADP molecule, producing ATP. Real-world applications involve anti-aging research. Nicotinamide adenine dinucleotide levels in the body decline with aging, and NAD precursor molecules are being researched as anti-aging health products. Watch the video from Crash Course Biology for more information on cellular respiration. Click the link below to explore the process of digestion and how it connects to metabolism and cellular respiration. Complete Part 3 of the Biomolecules on the Menu assignment using the link below.